Hello, everyone, and welcome to today's webinar, Automated Surveillance for Deep Characterization of the Tumor Microenvironment Using Ultra-High Content Imaging. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more, visit miltenyibiotec.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speakers, Dr. Telma Santos, Global Product Manager for Ultra-High Content Imaging at Miltenyi Biotec, and Dr. Nizar Yousif, Global Product Manager, Immunology, Immuno-Oncology at Miltenyi Biotec. Telma and Nizar, you may now begin your presentations.
Thank you very much, and thank you everyone for joining today's webinar. I am Dr. Telma Santos, and together with my colleague Dr. Nizar Yousif, we will be presenting our webinar named Automated Surveillance for Deep Characterization of the Tumor Microenvironment Using Ultra-High Content Imaging. And I hope you can appreciate this very nice image that we made. It's actually made with a composite of a real sample that you actually will see later, and the background has just been changed to this very nice teal color, but all over it's still a real microscopy image. And we will be diving, of course, in a more scientific talk, but it's also very nice, and you can, with your science, produce quite the piece of art. So I will briefly introduce the agenda for today. I will start with a short introduction. I will talk a little bit about our technology and our imaging platform components. Then my colleague will deep dive into the scientific aspect of it and will talk more about the importance of ultra-high content imaging in the TME research, and will show you a couple of data from different types of cancer, and we will end up with a very nice summary before we can take some questions. I will not go too deep into this part, but I couldn't start without just referring to the tumor microenvironment, which is at the center of our talk. As we all know, the environment around the tumor is quite heterogeneous. We all know that, but we still cannot fathom exactly how heterogeneous it is. We have a guess, but it is clear that we need a vast range set of tools that can allow us to study this complexity at this level of complexity. Here I'm showing you what is the gold standard methods in at least pathology. We have the hematoxylin used in staining, which is used quite often to determine what the sample looks like. We have chromogenic immunohistochemistry that helps you select a specific marker and see it. And we use also now more and more immunofluorescence, as you can see here in this nice image of a tonsil, of a human tonsil. But as you can see, because you would recognize that a composite has normally this kind of colors, we often do 3-plex, 4-plex, sometimes pushing it to show different markers. But this is quite limited. We always need some markers that are the standard ones. And if we want to go deeper into describing new ones or new combinations, this can get very complicated very fast. But why is it that we are limited? When it comes to immunofluorescence, we are limited actually by the light spectrum and the properties of our fluorophores. they overlap often, although we have a lot of available fluorophores. They often overlap and makes it very difficult, almost impossible to distinguish them. Therefore, just using them at the same time is not really an option. But what if we were not limited by the spectrum, because we would just simply use the same fluorochromes or set of fluorochromes in each cycle again and again? In this way, we could have so many markers, right? And that is exactly what the mixed technology is about. It stands for MACSima Imaging Cyclic Staining, and does exactly that. And it is as classic and elegant as you imagine. It's that simple. The mixed technology happens in the MACSima Imaging Platform. This is an instrument we developed at Miltenyi, and basically what it does is that it iteratively does cyclic staining. It stains the sample, images the sample, and erases it. But let me show you a bit more how it works in an easy way. Basically, take your sample, your fixed sample, and this is important. You fix your sample, you prepare your sample, and you put it in the MACSima. You take your reagents and you put them in a plate prepared so the MACSima can pipette it. And then you'll have a sample that is unstained. And then the MACSima will pipette to your antibody or antibody cocktail, take a picture, and then erase the signal. And therefore, after erasure, we can start all over again, just like at the first cycle. You stain it with another set of antibodies, you image it, you erase it, etc., and so on and so forth. And just that way, you can do it a very high amount of times. Actually, we don't even know what is the limit. You can continue as long as you have antibodies actually to stain with. Our current record was 400 because we had 400 in our hands. And it's quite impressive, I must say. So let me show it just one way of how it looks like. So here you have a sample of breast cancer that my colleague will talk about a bit more in detail later. And here you can I hope you can appreciate how amazing it is to have just this amount of different markers in a single sample, and how how such of a breakthrough this is to be able to have so many markers now. And of course, my colleague will be a bit more deep into what it means scientifically speaking. And now, just to end, I would like to just finish by explaining a bit more what we mean when we say an imaging platform. The MACSima Imaging Platform does not refer only to these quite pretty, I must say, instruments. It is a full solution. This is what we want to do, is to provide a full solution here. And when you have a MACSima instrument, we also have available at Miltenyi sample carriers that can accommodate a vast range of different samples. So we have here our Maxwell imaging frames. These frames have different sizes for the cavity reaction, and here you can mount any kind of a very basic or standard microscope slide with your fixed sample on it. And then you'll be able to insert it in the machine and then do the cyclic staining. But of course, if you also do not work with tissue but cells, we have the option of the Maxwell 24 here, where you can cultivate your cells, fix them regularly, and then have them in the machine also to perform cyclic staining. Very soon, we will also get a range of what we call our microcavities families, Maxwell microcavities. We will have them in different formats, one that is a slide format that can be used in the Maxwell, and also a plate format. And these disposables basically have millions of microcavities that can accommodate a cell, and this is perfect for cell suspension studies. Besides having the what you need from your sample, at Miltenyi we also thrive to have for you the best antibodies, the best reagents. And we have actually a very big range of mixed validated antibodies. And what we mean by that is that these antibodies have been tested in this technology already, so they are already validated. We also offer a couple of different technologies. We have different erasures mechanisms, and one part is the Realise and Readylise technology, which is our gentle signal erasure mechanism. And we also have these pre-made plates, antibody plates, dried reagent plates that you don't even need to pipette your antibodies. You basically put it in the machine, and the machine will reconstitute the dried antibodies and will pipette them into your sample, reducing even more how much you have to pipette before starting your experiment. It is also important to just mention that although we have a vast range of mixed validated antibody, this is not a completely closed platform. Because of the classical nature of our technology, being that our antibodies are linked to fluorophores, our primary conjugated with fluorophores, it is quite easy for someone to just use external antibodies, given if they meet our certain dependencies for it, which are not very difficult. But the important part is that we have antibodies, but you can also use different ones. This is not a closed system. And last but not the least, I just wanted to mention that with the MACSima Imaging Platform also comes a very good image analysis software. This is not just something that was added with a couple of add-ons, but this can really, really make your research complete. It's from A to Z. You have your sample, you start with a sample and a set of antibodies, and you can end up with real results. This is a software that was specifically made for the MACSima data, that can handle so many markers, that can do a very big set of analysis. We have analysis tools that are state-of-the-art, and of course, it is highly intuitive, interactive, and I would really advise to contact us if you want to have a look, because this is something from another world. But I might sound biased here, but really, this is a great product, and it's a family, it's a full solution for you. And I will end with my my part of explaining the technology, and my colleague will now explain to you a bit more the scientific part, of course, what you can get out of this, because this is not just showing the technology, but it's for you to understand how it can help in your research.
Thank you very much, Telma, and hello everyone from my side. Now I will give you a brief introduction about the tumor microenvironment, and then I will present some data from different types of cancer generated by the MACSima. So what does the tumor microenvironment mean? Well, the tumor microenvironment is a very busy traffic in which many different components sit, such as cancer cells, immune cells. These are immune cells that are resident in the tissue, or infiltrating immune cells. And these are coming from outside the tumor, for example, from the bloodstream. And in the TME, we also have extracellular matrix proteins and several secreted factors. On the left side, you observe a schematic of proposed mechanism of action of normalization strategies of the TME to improve immunotherapy. Combined administration of immunotherapy with stroma and or vascular normalization restores vascular functionality and alleviates solid stress. leading to improved tumor perfusion and oxygenation, skewing tumor-associated macrophages, TAMs, polarization away from the immunosuppressive M2-like phenotype to the M1-like phenotypes. These are macrophages with pro-inflammatory phenotype, then boosting immunity, for example, CD4 positive and CD8 positive T cells, and thus enhancing cancer cell killing. So the biological process occurring in the tumor microenvironment play an important role in tumor development and do affect the clinical outcomes of cancer patients. In this table on the right, you see that effects of the immune infiltrate on the prognosis of patients with cancer. And you also see the relevances of CD8 positive T cells, tertiary lymphoid structures, TLS, regulatory T cells, Tregs, CD68 positive macrophages, and more specifically macrophages of an M1 or M2 subtype to overall survival outcomes. So the presence of CD8 T cells, TLS, as well as M1 macrophages in the TME is a good sign and indicates positive prognosis. However, the detection of M2 macrophages, CD68 macrophages, as well as Tregs is in most of the cases associated with negative prognosis. Now, let's have a look at some example of pain points that make TME research challenging. The TME consists of many cell types and many structures, and this makes it difficult to understand what is going on there. There are lots of complex interactions and processes going on in the TME. It's very important to understand TME heterogeneity. We need to know where is the location of these interactions and processes, so the spatial distribution is important. The MACSima and its mixed technology offer you amazing options to overcome many of these challenges. Now, I would like to show you some examples of data generated using the MACSima and the mixed technology. This is about the characterization of the tertiary lymphoid structures in the tumor microenvironment. But before I show you data, let me say some few words about the TLS. This cartoon shows a TLS located within a tumor. Please see maximize view in the middle of the slide with a CD3 positive T cell zone containing DC lysosome associated membrane glycoprotein, abbreviated DC LAMB, with dendritic cells and fibroblastic reticular cells, and a CD20 B cell zone with a germinal center. Plasma cells, antibodies forming immune complexes with tumor antigens, and follicular disease. The coordinated actions of CD8 positive cytotoxic effector T cells and B cells generated in TLS enable in situ tumor destruction via direct tumor cell killing, the so-called antibody-dependent cellular cytotoxicity, mediated by macrophages and or natural killer cells, and local complement activation. So central memory T and B cells generated in TLS circulate and protect against metastasis. In some cancers, TLS located in inflamed regions adjacent to a tumor may serve as a niche for tumor cell progenitors or tumor. So the TLS is a very important structure in the tumor microenvironment. Now, I would like to present you the first story where the TLS was investigated in ovarian cancer sample. You see the tumor tissue represented by the EpCAM segment in green and the associated leukocyte marked by CD45 in red. The nuclei are depicted in blue and visits are shown by the CD31 signal. The massive accumulation of leukocyte adjacent to the tumor already indicates that this could be TLS. But we can dig a little bit deeper and have a look at more compositions of the TLS structure, which can further support this data. In this image, you see B cells distribution in the structure. B cells are shown in purple as CD20 signal, and the B cell signal appears as a zone, which is, as mentioned earlier, one of the TLS characteristics. But where are the T cells? In this image, you observe an overlay of 7 markers indicating the tumor cells in green, various immune cells, including B cells in purple stained with CD20, plasma cells in white stained with CD138, myeloid cells in yellow stained with CD14, and endothelial cells in red stained with MACA79, which is a marker of high endothelial venues, abbreviated as HEVs. This data was generated by our research and development using the MACSima, showing biomarker screening in pancreatic ductal adenocarcinoma. In each image, you observe an overlay of 3 markers, including DAPI in blue, EpCAM in red, representing the tumor signal, and the marker of interest shown as green signal. The marker specific name is written in white on each image, and here they initially tested 97 markers, but unfortunately 4 markers did not show any signal. And I think we can appreciate how much valuable data we can obtain using this technology. This result is collected from the recent brilliant work published in Nature Communications by our research and development colleagues. The idea behind this work was to identify CAR T cell target candidates in pancreatic adenocarcinoma using different methodologies. Among them, the MACSima and its mixed technology allowed them to generate fascinating data and helped the identification of specific CAR T targets. So in this result, the cytokeratin staining is shown in green, which is showing the tumor signal, the DAPI in blue, and the markers of interest are stained red. And among all candidates, they selected the markers that are co-expressed with the tumor cells, which means that they selected the red signal that are co-expressed with the green signal. So these markers were only specific for the tumor, and the perfect candidates they selected are the ones that are surrounded by a red square here. These are CLA, CD66, and CD318. This result is from the same study. Here they took advantage again of the MACSima platform. They looked at CAR T cell infiltration on tumors treated with either CD66C or with CD318 CAR T cells. You should see the CAR T cells in purple as they are stained with LNGFR, which is the CAR T cell reporter marker. CD318 CAR T cells infiltrated the tumor and did a great job, and this resulted in less tumor presence. And combined with the observation of CAR T cell infiltration in case of CD318 treated group, they observed CD68 macrophage infiltration, which is really interesting observation. And in the paper, they performed validation experiments which confirmed these findings. In my last story, I would like to show you the brand new data generated from breast cancer sample. We started by looking at different cell populations. Here, the tumor cell is shown in cyan color. the fibroblast in red and the nuclei standing dappy in blue. Here we observe a wide distribution of the fibroblast outside the tumor as well as in between the tumor. Here we detected T cell signal in red, however, mostly distributed outside the tumor. But we also observe some relatively few T cells close to the tumor. In this image, we observe that some of the T cells are actually CD8 positive T cells, overlay signal in yellow. Here we looked at HLA-DR signal. The presence of the HLA-DR signal in this image proves the existence of MHC class II receptors, which is also one of the TLS characteristic features. Interestingly, in this breast cancer sample, we were able to detect anti-apoptotic signal. This is BCL2 signal depicted in red outside and within the tumor. In fact, the role of BCL2 in restricting apoptosis and its widespread expression in breast cancer has attracted attention as a therapeutic target for many years. We also observe a key 67 signal shown here in green, indicating cell proliferation. Now, I would like to wrap up by summarizing the benefits of the MACSima and its mixed technology for TME research. Using this technology, you can do comprehensive characterization of the tumor microenvironment components and also understand their spatial distribution without having to worry about any marker limitations. If you are interested in identifying tumor-specific targets for cellular therapies, then you have an amazing solution. Of course, with complex investigations comes complex amount of data to analyze. Here you can rely on the QI tissue image analysis software for the MACSima. There are several amazing analysis options you can use in this software. For detailed tissue analysis options using this software, please watch the on-demand webinar highlighting the benefits of the MACSima in lung cancer presented by my colleague Dr. Santos. We know that human tumor samples are precious and sample availability for research is in many cases a challenge. So through this technology, you can obtain large amount of data from a single tissue section. And finally, and most importantly, our internal experts at Miltenyi Biotec are ready to guide you throughout the whole ultra-high content imaging workflow in your TME research. And this is our disclaimer. Thank you very much for your attention. And now we are happy to take your questions.
Thank you, Telma and Nizar, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, how is the stripping step performed? Is this automatically handled? Any signal loss after repeated stripping?
This is a good question, actually. No, the process does not include stripping. So the signal is erased by photo bleaching using our fluorophores, and another erase mechanism this technology uses is controlled fluorochrome release of the fluorophore using the Realize technology, which is developed by Miltenyi. So this is an automatic process done by the machine. As soon as the experiment starts, you don't need to do anything. Yeah, so just relax and fun.
Thank you, Nizar. The next question, is there a limit for the number of markers? And how much can we go? How many can we do?
Our record is 400 markers, yeah, and we are testing more in-house to break this record. So please stay tuned to get the results of the next race.
Great, thank you, Nizar. Our next question, what is the largest field of view and what is the best resolution possible? Cellular, subcellular? Lots of questions there.
Hi, I will take care of this one now. So the field of view is defined by our camera, but we actually can do several regions of interest with a lot of fields of view. Our field of view is about 500 microns per 250, but there is no limitation the amount of field of views or ROIs that you can do. It's basically the size of your sample. So of course the maximum would be just slightly under a full slide, but that is what we can do right now. The resolution, sorry, I forgot the resolution. So the resolution, the best possible is around the 425 nanometers, and it's enough for subcellular resolution.
Great, thank you, Telma. The next question, can you elaborate a little bit about the options for data quantification? How user-friendly is the MACSima QI software?
Thank you. I'll take also this one. The QI software, which will be officially released very soon, is very, very nice to use. It has been tailor-made for actually this kind of data. It's super interactive. So I would invite people who are interested in the MACSima, but also in the QIT sheet, to always contact us, and we will have a pleasure to show you or have you try it. But we also have available on our website some videos about it, so feel free to check it on and ask us for whatever you need.
Thanks again, Telma. The next question, are the antibodies available from you or collaborating vendors?
Okay, so I'll take this one. So we have a big amount of antibodies that are tested and validated for the use within the MACSima software instrument. But of course, it's also possible to use antibodies from other vendors. Basically, you just need to fulfill some basic requirements, which are being primarily conjugated to one of our photobleachable fluorophores, so CP and APC, and be sure that you have a desired staining within 10 minutes of incubation, because that is how the process works within the machine. If antibodies fulfill these requirements, you are free to use also antibodies from other companies.
Thank you, Telma. Our next question. Let's see. Would it be possible to image organotypic slices with 350 nanometer thickness, or micrometer, sorry, thickness, using the MACSima?
I'll go for this question. Yeah, the MACSima imaging technology is based on wide field fluorescence microscopy. So for the best results, yeah, of course, thin sections are preferable. So from 3 up to 10 micrometer. And in our experiments, we averagely use 5 micrometer sections. Yeah.
Thank you, Nizar. Our next question. Can a process slide be stored and analyzed with new markers, and for how long?
So a process slide should be able to be stored and analyzed with new markers. You just have to be aware of the conditions of your slide, of course. So be aware that we do not remove the antibodies from the sample, so you in principle cannot use the same markers again. And of course, a sample that is within a buffer can, depending on the quality of the sample, last more or less. So it really depends on your protocols of maintaining your sample. But in principle, this is possible, yes. The how long, I would say in general, because if you use a PFA or an FFP that has already been retrieved, I would say a couple of weeks to be on the safe side. But as we know, scientists tend to test these limits themselves, and this is very, very dependent on the sample and the quality of the sample that you're dealing with.
Thank you, Telma. Another question, what type of data output does the imaging analysis software generate?
So that's a great question. So QI-tissue will analyze your images and will have a quantification after segmentation based on your cells, or if you want to do a kind of image segmentation based on pixel sizes, you can also. So we have outputs as regular CSV files, and you can also have the raw data output as FCS files that can be read in some flow, in most of the flow softwares. So this is the numerical data that comes out of QI-tissue, but you can also, of course, out of the MACSima, we generate regular OMITIF files. So any image analysis software will be, in principle, able to read this kind of data as well.
Thank you again, Telma. The next question, can I use my own antibodies, or do I have to use special antibodies with the MACSima?
I think my colleague Telma touched that question already, but I can elaborate again. Yeah, that's not a problem at all. So you are free to use your own antibodies as long as they fulfill the requirements of the mixed technology. So meaning that the antibodies should be primary antibodies conjugated to a bleachable fluorochrome, and they should be able to stain with a good signal in 10 minutes at room temperature, so which suits their MACSima staining mechanism. And in-house, we used our own mixed validated antibodies, yeah, but no, it's not restricted to Miltenyi mixed validated antibodies at all.
Thank you, Nizar. Yes, I think we had two very similar questions, so I really appreciate your answer. The next question, how many samples can be analyzed at once?
So this kind of sorry for the echo. So this the amount of sample depends clearly on your reagents, actually. So it really depends on your reagents, what kind of chamber, and the amount that you have. But we currently are more or less limited to one slide, but we focus primarily on the amount of markers that you can do in one sample. But just a side note, we are thinking this instrument to be able in the future to analyze more, of course. But the primarily goal of the MACSima is to analyze a high amount of numbers in one single sample.
Thank you, Telma. Our next question, does the MICS technology have preference to specific type of fixation?
Actually, the mixed technology has nothing to do with the fixation, but of course, your antibodies need to be compatible with the fixation you are using. We have a large imaging antibody portfolio that is compatible with all types of fixation, whether it's FFPE or PFA or acid on fixed tissue or cells, so whether it's from human or mouse samples. Yeah, and you can check that in our website if you're interested.
Thank you, Nizar. I want to clarify, it's mixed technology. I mispronounced it, so I apologize for that. Next question. Can you simultaneously run an experiment and analyze a previous one? Are data transferred from one to the other as they are produced or at the end of the experiment?
So normally the data will be available as it is made, but of course, it depends on your kind of analysis. If you need to wait for all the markers to start your analysis, you will have to wait for the end of the run. But in principle, you can start analyzing if you wish, for example, to start, for example, the segmentation parameters that can take a long time. You could, in principle, do it. The MACSima is equipped with 2 computers, so in principle, you will have one computer dedicated to the experiment run and one to the analysis. So this should be possible without really affecting the experiment.
Great, thank you, Telma. The next question looks like a follow-up question. So output data is quantitative?
Yes, I remember that question. Yeah, yes, so it's quantitative. Yes, we have a list of very usual features related to the fluorescence, but also the shape and the location of the cells after segmentation. So all of this is quantitative and can be analyzed in different ways and manners, but it's a very quantitative way for sure.
Great, thank you so much for that clarification, Telma. Our next question, how does the technology cope with autofluorescent tissues?
That is again a great question. I think that if we have in the audience people who are used to deal with FFP fixed tissue, we all know that autofluorescence can be a pain also in some kind of tissue, specifically like the lung or even the brain. It can be also a bit difficult. So there is an initial step when we do the run that will do a long photo bleaching to ensure that the autofluorescence is removed. But we have to say that the images are acquired always as a signal image, and we also acquire after signal erasure in every cycle. And when we process the images to stitch them together and to register everything, we also do a step where we remove the signal from the last if you want, erasure signal. This way we are removing most of the signal that is still maintained, that can come from autofluorescence or anything that might be got after it. But you have access to all of these images, so you can follow up how good the run has been. But in principle, when you have your images ready to analyze, all the autofluorescence and some background signal has been removed.
Thank you, Telma. Our next question, how can you ensure that the signal from one cycle is not detected in the subsequent cycle in case the erasure is not 100% efficient?
Yeah, so this comes a bit as a continuing part of what I said before. So because we always take pictures in between cycles after a signal erasure, we make sure when we're processing the images that we are removing the signal that might still be there because of autofluorescence. And this way we are sure that the images that come out after processing are really ready to be analyzed without all these issues.
Thanks again, Telma. It looks like we have time for one more question. Is photo bleaching damaging to the sample?
No, not at all, yeah. So it's a very gentle process, yeah. So exactly, you don't need to worry about, sample damage, yeah.
Thanks for that answer, Nizar. Appreciate that. And thank you both, Telma and Nizar. Do you have any final comments for the audience today?
I would like to thank everyone who assisted to this webinar today. It was a pleasure to also answer your questions. I hope that we shed some light on our technology. Just wanted to say that you have available on our website a lot of information. And of course, if you have a bit more of interest, do not hesitate in contacting us. We will be more than happy to guide you through this technology if you need to. Nizar, do you want that?
Yeah, and thank you very much. Also from my side, thank you for your attention, and I wish all of you a great day. I'll see you next time.
Fantastic. Thank you again, Telma and Nizar, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.